Today's lecture is streams for visually guided actions. So we saw way back here in lecture 3 that the visual stream divided into two parts, a what stream and a where stream. And so today we'll look at that where stream. So we saw that information flowed through V1, V2, V3 up here to the parietal lobe, and then down here the inferior temporal lobe. And this is where the actions are coded. So it's the representation of where things are around you. And if you look at the links to today's lecture, there's a good video of a patient that has optic ataxia, that is, a patient who has trouble reaching for things in space. Now, so this area here that we're look at today is involved in guiding all kinds of movements. They're the movements of the eye. Ones we'll be interested in are called Cycads, because they're very fast movements of the eye. Also grasps and reaches. Now grasps and reaches are different because in grasping you actually put your hand around the object, where a reach is sort of more of a pointed movement. And then feeding, which is a very important movement. And all these have different effectors, of which you're largely unconscious. When you're playing soccer, for example, you're going to hit the ball unconsciously with either your left hand, your right hand, your head, or your legs. Now we saw that the what stream was sensitive to these optical illusions. So this line is of the same length of this line, but because we have this sense of perspective, we assume that anything that's over here is longer because it's farther away. So we assume it's a longer bar than this bar, but if you actually measure them, they're of equal lengths. And so for the action stream, it's important to point to or reach for objects exactly where they are and not be fooled by these illusions. And in fact, if it's working right, it won't be fooled by these illusions. So the movements will be accurate. Now, we saw that each of these streams used different coordinate systems. The one we'll study here works on the coordinate system relative to your body. And again, that part of the body can depends on what type of movement we're going to consider. We'll see that there's each type of movement has perhaps a different egocentric frame. We'll see, for example, that the retina works, so if you're coding where your eye should move to make a Cycad, you work in terms of where it is on the retina. If, on the other hand, you're trying to locate something based on what you hear, you're working with respect to things around your head. And finally, if you're making a finger movement, you're coding things with respect to your body. Now, patients with lesions of this area have trouble both making eye movements and grasps and reaches and feeding accurately. These areas that we'll be looking at are concentrated around this interparietal sulcus, here within the parietal lobe. And again, this is a good place for these locations to be coded, because they can be coded on the basis of touch, or on the basis of hearing, or on the basis of vision. Now, we'll go through several of these areas. One of these is called the Parietal Eye field, and it represents things with respect to the eye, the retinotopic locations. And neurons here respond both to vision and to auditory stimuli. This one here, the parietal arm field, involves reaching movements, and it is involved in you reach to locations within something called the immediate peripersonal space, and that's basically defined as those spots that you can reach to. And here again, neurons are visual or touch. And here especially you get this optic ataxia, which is with difficulty in reaching movements. Now, there's another area that's similar but different. It's called the parietal grasp field, and it's involved in picking things up. So how you pick things up really depends on what the object is. If it's something like this strawberry here, you have to pick it up gently. If it's something like a knife, you have to make sure you pick it up by its handle. So that means there must be information coming from the Watt stream to this area that helps this area define the object part of what you see. And lastly, there's an area called the parietal face field, and that's involved in things like eating. Here I am bringing the strawberry up to me and taking a bite. And it's involved in not only feeding, but things like kissing. So you have visual input and also tactile input coming to this area. Now, each of these areas represent different parts of space. The parietal face field is something here close to the face. They represents locations on your face. The objects could be far away and heading for these different locations, but they're always locations on your face. A grasp field will be the area that your hand can grasp. The parietal arm field will be this whatever you can reach to, this peripersonal space. And the parietal eye field is all the areas that you could look at, which could be quite a distance even further. Now, let's look for a minute at the parietal face field. Again, as I mentioned, it basically defines areas on your face. And so, for example, here, this particular neuron will be sensitive to something touching the lower lip here. It's also sensitive to things that are heading for this lower lip. Okay, so they don't have to touch it, but the direction this object is moving seems to be towards this lower lip. And it could be moving from any direction to this lower lip. Doesn't matter from where it comes from, as long as it has its final point is the same thing. And also surprisingly, it doesn't matter where the eye is pointing. Okay? So the eye can be looking up or looking down. It can still tell that this object's heading for the lower lip. It can take into account where the eye is pointing and determine that what the eye sees, even though the eye sees something totally different when it's looking up from when it's looking down, the brain, this area can still compute that the same point in space is going to hit, and this neuron is going to fire. And there's other neurons, okay, that do the same thing, but for some other point on the face. Okay, now let's look at, review again some of the pathways for making these Cycads, the visual grasp reflex, that we began with, I think it was either lecture 1 or lecture 2. And there's a pathway from the eye to the superior colliculus, and from there to the centers that control the movement of the eye. Okay? So this area is activated by something unexpected flashing somewhere in your periphery. And if something like that happens, one of you raises your hand, that'll signal this pathway, and I will look to, hopefully, to whoever it is that has his or her hand raised. Now, there's another pathway that has a longer route, and it goes through to the visual cortex, through this parietal eye field that we spoke earlier about, then to the prefrontal lobe, and then to the frontal eye field, which is a structure similar to the superior colliculus. It too generates eye movement. It does so by sending a signal to the superior colliculus. Okay, so that can generate a movement to something that you remember you saw in this location, because this prefrontal cortex is where your working memory lies. And so I can look to my mouse because I remember where it was. Now, to look to something, you can imagine that on the superior colliculus, this is both sides of the superior colliculus, the left and the right in one diagram. You can imagine the fovea here is down the middle. Notice, unlike the visual cortex, very little of the fovea is represented in the superior colliculus, and it's mostly the periphery that's represented, because you want to elicit visual grasp reflex for things that aren't on your fovea. So, but again, these neurons here have large receptive fields, and so it might be one here that's firing a little less, and one over here a little more, and one here right in the middle that's firing the most. And all these neurons firing can be described as a hill of activity. Okay? So it's an area, and the height represents how much. And so this point here, even though each one doesn't code very accurately where the object is, together, by looking at where the peak of this activity is, the colliculus can code locations quite accurately. And from there it makes a movement of the eye. Now, one of the things is that when you see something in your periphery, a hill of activity appears in your periphery, and then you make a Cycad, and a hill of activity then appears in your fovea, because you're looking at that object. Now, before, when you've got a hill of activity in the fovea, there's something that inhibits more saccades from happening. So through this circuit here, if the signal's coming from the fovea, you remain fixated. And when an object appears in your periphery, its activity has got to first reduce the activity at the fovea before this movement can begin. So you have to suppress activity in your fovea and then elicit a saccade to this peripheral object. So this has to be, in this way you're not distracted by sort of little movements that might, you don't look at every little thing that somebody's scratching their heads or typing on the computer. It has to be something that is unusual to attract your attention. Now, all these structures that we looked at, the retina, V1, you can all consider them as topographic maps. So they code locations in space. We saw the fovea. This is the fovea. It has a small representation in the retina, but because of the optic nerve concentration and in V1, it has a large representation in V1. But in the superior colliculus and these other areas, the parietal eye field and the frontal eye field, it gets back to a very small representation. And again, all these structures are interested in what's going on in your peripheral retina because it wants to direct eye movements to those locations. Now, again, if you stimulate this area or this area, you get a saccade to one or the other area. If you stimulate both at the same time, the movements cancel each other. Again, to remind you that I've drawn these as one map, but they're on different halves of the brain. So there's a midline that runs through all of them, and half the fovea is represented on one side of the brain, and half the fovea, and the rest of the retina is represented on the other side of the brain. Okay, now let's compare the activity in these areas when you're performing one of 4 different types of everyday movements. The first one is when something an object appears in your retina, and then it appears in your V1, but the task is to simply continue looking forward and not be distracted by this object, okay? So what will happen there? It'll mean that the parietal eye field, which is for saccades, the location of things that you attend to, your attention is focused there, that won't be activated. So again, attention won't let activity pass through these higher areas, and only V1 will be activated. So all you see is the activity appearing in V1. So A happens here, and on the contra V1 you see a hill of activity. Now in this case, what you're asked to do is the stimulus appears, and you're asked to look, to make a saccade to the thing that appears. So here it appears on the retina and the V1, then the parietal eye field. Now attention is focused to it. Then over here, a hill of activity occurs in your frontal eye field, and you make a saccade, and the hill appears in your fovea. Now, in this third condition, or not third condition, this test condition, okay, we have two objects appear, and I'm going to go through these possibilities one by one, and you tell me whether or not there'll be a hill of activity in either of these locations. So the task is, it appears at A and B, but you as a subject is required to make a saccade only to B. Okay? So you're asked to only attend to this object. Okay? So will you see activity in the right V1? Yes? Okay. Left V1? Yes? Okay. Right PEF? What do you think? No. Okay. We won't put the check mark here. Left PEF? Yes, because that's, you want to make a saccade to B, and that's on this visual field, so it's going to be represented over here. And then right FEF? No. Left FEF. Good. And that's indeed what will happen. So you see these hills of activity appearing in all those locations. You then look to B, and again there's activity in the fovea after the movement is completed. Okay. So we saw that if only a visual stimulus appears, there's activity without attention, then only the fovea, the V1 gets activated. If there's attention, then we get activity in the frontal eye field, and if there's a saccade, we get an activity here in the FEF. But what if, rather than make a saccade to it, we're asked to point to this A? Okay. So attention is directed to A, so you'll have activity both in the parietal eye field and in the parietal arm field. But because you're not asked to make a saccade to there, your fixation will be still straight ahead, but some other structure will initiate a pointing movement towards it. So in summary then, the retina and visual cortex requires only a visual stimulus. The parietal eye field requires the visual stimulus and attention, and this selects a target for a movement. The frontal eye field and the superior cortex requires an orienting response to be directed to the eyes, and in contrast, if it's a movement of the arms, then the parietal arm field is activated. Okay. Now let's look at what happens when you have memory. Okay. So now we see this briefly flash, and we have another structure here called the prefrontal cortex, where your working memory lies, which then maintains the memory of this object. So this object briefly flashes, and its memory then persists in your prefrontal cortex. So what happens if two objects flash, A and B? A and then B. Okay? So then two hills of activity then go away. So they appear here in the visual cortex, but disappear. But then these two hills appear for a more prolonged time in your parietal eye field and your prefrontal lobe. But if you maintain fixation, that is, not look at them, your frontal eye fields will only have activity in the fovea because you're maintaining fixation. But now what happens if you look to the object, look to where you remembered these two structures to be? Okay. When that happens, the memory of those objects gets shifted. As soon as you make the saccade to A, or the memory of A, okay, your eyes are pointed to where A was. So your fovea then is pointing to the object. So A then is located in where your fovea is. And so you have the activity that was over here in A moves from the fovea appears in the periphery here. A saccade is generated, and that hill moves back to the foveal representation. So here you can see these hills shifting over in your memory. Now, what causes that shift? What causes these locations to shift? Because your eye movement is shifted. If I remember that something was over here, and I look over here, even though there's nothing moving on my retina, just the memory of that, what moves it inside my head? Well, what moves it is a copy. Again, your quality discharge generates a signal that your eye is moving, and that signal moves this map, moves targets on this map, recodes them to a new position. Okay, so it moves them in all these positions, moves them in the parietal eye fields, and in the frontal eye fields. And so now if you wanted to generate a saccade to B, you could do so because it's moved now to a new location, and you correctly direct the saccade to where it is now. So it moves things to where they would be, were they visible. And so that's very useful because every time you move your eyes around, object gets shifted all over the place on your retina, and yet you know everyone in the room is staying still. So even though they're moving all over your retina, you have this sense of the world, people in this world are stationary. And the sense of objects being stationary, even though your eyes are moving and the slip is all over the place, is generated by quality discharge. They move where you expect the object to be to where you actually see it to be. So the two maps match. The one from the visual cortex matches that in your parietal frontal eye field, prefrontal cortex, the memory, and therefore in this parietal eye field as well. Okay, another topic, Waldo. Now, both the frontal eye fields and the parietal eye fields are involved in covert shift of attention. So that they're involved, we saw just the previous section, that they're involved in overt, that is, every time you make a saccade, that's an overt shift in attention. But they're also involved in covert. So you can maintain fixation here, and you can do these little shifts of attention. You can say, okay, I'm kind of interested in something over here by my mouse, okay? And you direct your attention to something over here, but you still maintain fixation straight ahead. Okay? So you explore this space around you, and you can do it very quickly because you're not making a movement, and you can look for possible positions where to make a movement. Where is Waldo? And if I think, Waldo is one of these places, this is a possible location for Waldo, then I'll make a saccade to Waldo, and I'll put Waldo in my fovea. Okay. So that way I can make these, I can avoid making these more time, slower movements of the eye with a saccade. So, we're finished quickly. So in conclusion then, in the next session we'll cover the somatosensory field. We'll see that it also has a map, and it had different places along this map code different places along your body. And so we saw today the parietal system has many maps. They're all locations of peripersonal space. And I said that this is the area space that you can reach to. And then we can reach to either hand or your feet. But you can extend this space by taking up things like a stick. If you have a stick, that representation in your cortex gets bigger. And if you drive things like a car, and with practice, you get to expand that space to the bumpers of the car. And so you can drive this car and park it exactly where it should be without bumping the cars in front of you. It becomes like part of your own body, your peripersonal space. Okay, that's it. Thank you.